Hello and welcome to today's Revive webinar by the Global Antibiotic Research and Development Partnership GARD-P. My name is Astrid Pence-Moore and I'm hosting today's webinar on discovering and developing new treatments for tuberculosis. We had the pleasure to collaboratively organize this webinar together with the TB Alliance for today's World TB Day. And I already want to take the opportunity to thank our collaborators and especially our speaker. Revive is GARD-P's education and outreach program, and it aims to connect and support the antimicrobial R&D community by facilitating learning, connecting people, and sharing knowledge. Our webinars are part of our educational activities, and all webinars are recorded in full and can be viewed on our website, revive.gardp.org/webinars after the live broadcast. On our website, you also find other content, such as our antimicrobial viewpoints, which are articles by leading internal and external experts on various topics related to AMR. And we have also, end of last year, launched our antimicrobial encyclopedia, which provides short definitions for key terms in the field. At the moment, we have about 75 terms there. Some of them also have short videos by experts. And in the coming weeks, we will upload more than 50 new terms. The presentation today will be followed by a Q&A session, as always. And I invite you to submit your questions in the questions window, as shown on this slide. You can do so at any time during the webinar. You don't have to wait until the presentation is over, but we will only respond to the questions, or the speaker will only respond to the questions after the presentation. So today's speaker is Nader Fotouhi. He is the Chief Scientific Officer of the TB Alliance. And our moderator is Lydia Nakiyingi. She's an infectious diseases physician, research scientist, and senior lecturer at the Makerere University College of Health Sciences in Kampala, Uganda. Welcome, Lydia. Please join us now with your webcam. Great, welcome. I will now hand over to you so that you can further introduce our speaker. Thank you very much.
Thank you, Astrid, for the introduction. Good afternoon, ladies and gentlemen. I would once again want to welcome you to yet another interesting webinar today as we commemorate the World TB Day. As we are all aware of the issues with the existing TB therapy, including the long duration of treatment, which is quite expensive and which may not be adequate in certain populations, especially those with HIV. So therefore, today's topic on developing new treatment for TB comes in quite handy to discuss and address some of these issues. So colleagues, allow me to introduce our speaker for today, Dr. Nader Fotouhi. Dr. Nader is a Chief Scientific Officer for the TB Alliance, where he guides and oversees the organization's research and preclinical development activities. He has over 24 years of experience in pharmaceutical industry, and he has significant research and expertise in a variety of therapeutic areas. Prior to joining the TB Alliance, Nader held various leadership positions at Hoffman La Roche. He was head of Discovery Chemistry Group at the Nutley, New Jersey site. He was the global head of the Discovery Technologies, and he has also served as the Nutley, New Jersey Pharma Research and Early Development Site Leader. Nader holds a PhD and postdoctoral fellowship in organic chemistry from Massachusetts Institute of Technology. He has authored and co-authored several publications and has given several presentations. Nader also has over 20 patents. So colleagues, let's welcome Dr. Nader for today's presentation. Dr. Nader, over to you. Thank you.
Hello, and thank you, Astrid, Lydia, for a great introduction, generous introduction. And thank you for giving me the opportunity today to tell you a little bit about TB Alliance, our mission, our goals, and some of our achievements, and end with future vision for how we can treat TB in a much shorter time frame. I believe I have control of the slides. Okay. So TB Alliance is, if you see the next slide, is a non-for-profit organization, and it's dedicated solely to discovering and developing and delivering better faster acting and affordable TB drugs that are available to those in need. It was founded over 20 years ago. We celebrated our 20th anniversary last year, and it started as a declaration of the Cape Town calls for TB Alliance to form a public-private partnership that was dedicated to developing new TB treatment. Now, most of you probably know this. TB is a pandemic. It is the leading infectious disease killer and a top 10 worldwide killer. Killing one person every 22 seconds, as you can imagine. Over 1 million children become ill with TB every year. 1.4 million people die from TB every year, and 10 million new cases develop. And it is a leader killer of people with HIV/AIDS, and more importantly, and it's more scary, the drug resistance is on the rise with about 500,000 cases annually. And it's something that we have always aimed as well at the get-go to address, and I will describe that a little bit later. Now, TB therapy currently, we believe, is obsolete. The arsenal of drugs were developed in the 1960s. I will have a slide later to make that point. The treatments are long, especially in drug-resistant TB, or 18 months or longer to achieve full treatments. Complicated. They can exceed 14,000 pills for drug-resistant TB. Expensive. Cost for treating drug-resistant TB can exceed $10,000 per treatment, and it is inadequate at this point in time. And the inadequacy is really for the long treatment, complication, and all those can lead to resistance because of non-compliance. And they can also be incompatible with some HIV treatment due to an unacceptable drug-drug interaction. So in TB Alliance over the last 20 years, we have really developed a new treatment at this point for highly drug-resistant TB, which will be a part of the focus of my talk. We've launched improved treatments for children with TB through formulation work. We've transformed how TB treatments are developed, and in that respect, our goal from the beginning, or our vision from the beginning, was to really focus on novel regimens, not just a novel molecule to be added on top of existing therapy, but really a novel regimen with new drugs without pre-existing resistance at the get-go, and really trying to limit ourselves to oral therapy as well. To achieve that, we've had to revive the pipeline of new TB drugs to have access to new drugs rather than some of the old ones that were developed in the 1960s. And we've done that through really mobilizing a global network of partners, and some of which you will see on a slide of our portfolio. And anything that we've developed and will develop and are developing will go along with a AAA mandate that we have, which is the regimen has to be affordable, available and adopted throughout the world. So our vision really for better TB medicines is it has to be simple, all oral, highly effective regimens. It has to be short. Our initial midterm goal is a 2 to 3 months treatment. And as I mentioned earlier, it has to be accessible and affordable to all people. And to achieve the maximum impact here, it really requires a sustainable pipeline of novel drugs, novel molecular entities, which would then form the basis of a universal regimen ultimately. And by universal regimen, we mean that it can treat all TB patients regardless of resistance. So drug sensitive, multi-drug resistance, and extremely drug resistant population. And it has to be, we have to also come up with an ultra short and effective therapy. And all of the therapies that we develop, all the regimens that we develop, have to have an appropriate formulation for children, rather than having to break pills and guess at the dosages that children need versus adults. So let me focus my attention first on drug-resistant TB, which is really becoming a major issue and potentially be devastating if we don't start treating it properly and coming up with novel treatments. So what are the challenges here? And really it's stemming from not adhering strictly to current treatments, which really leads to drug resistance. As I mentioned earlier, it's about 500,000 people, new cases come up every year. More than 50% of these cases are caused by direct transmission, drug resistant to drug resistant. Only 43% historical data of XDR TB treatments were successful. Drug resistant TB, as I mentioned also earlier, takes a long time, up to 2 years or longer. Take over 14,000 pills, in some cases injectables. It is estimated that by 2050, 75 million people could die or will die from drug-resistant TB. And about 1 in 3 of these deaths from antimicrobial resistance are due to drug-resistant TB. 1 in 3 of the AMRs. And it's estimated that the cost of the financial toll by 2050 could be in the order of $17 trillion. So what would be the benefits of new regimens for drug-resistant TB? Clearly, lower cost, save more lives, better outcome. As I mentioned, the XDR TB population, the best we could do was in the 40% range. Reduced health system burden, again, shorter treatment possibly, and higher cure rates. Fewer side effects, and we will discuss that also later. And more patient satisfaction, simple supply management, less pills, less number of pills, less new molecular entities, and then of course, ultimately reduced transmission. So this is an interesting graphics that really demonstrates the need for new drugs, the need for new regimens and therapies. As you can see, we had a huge effort in the 50s and the 60s, and a lot of great drugs came out of the research for the treatment of TB. And for a long time, this worked really well until resistance emerged. Even in regimens, it was known that if a single drug was given earlier on, like streptomycin, very quickly resistance emerged. But even in the 60s, once we discovered the current standard of care, years after, we realized that resistance can occur again due to poor compliance because the therapy was long, with a large number of pills to be taken at a given time. So as you can see, there was a dearth of activities here, and really no new drugs came out for a long time to support our vision and our mission of starting from new molecular entities to create novel regimens. In 1998, we saw Rifapentine come up, which was a derivative of rifampicin. But of course, Rifapentine, being similar molecule to rifampicin, was fraught with the same issues of resistance, of course, as rifampicin. And it wasn't until 2010 where we started finally seeing some new molecules being approved and really providing new compounds that we could then test and put in novel regimens. And the first one was bedaquiline, that we worked together with Janssen, and that was registered in 2012, or approved in 2012. 2 years later, a nitroimidazole delaminate was approved by Otsuka in 2014. And as you probably know, through the BPAL regimen, Pertominate was approved in the regimen with bedaquiline and linezolid for extremely drug-resistant TB. And I will describe that later on in the presentation. And very recently, a new regimen, an optimized regimen that included moxifloxacin with the standard of care, reduced the 6-month therapy to 4 months therapy. So we are really in a good position to start developing novel regimens to be able to address the needs of this disease. And to achieve that, as I mentioned, we need new compounds, new novel molecules that really don't have any pre-existing resistance at the outset. And I'm showing you the portfolio that we have developed, and this is just our portfolio, and there are a number of other organizations and institutions that also are starting to develop novel compounds. But as you can see, we have a large number of projects in early phases of research in lead identification, a good amount of projects in the lead optimization that ultimately will allow us to select novel compounds or novel clinical candidates to go into preclinical development. We have 3 compounds now in phase 1. I will describe those to you shortly. And we have 2 compounds in phase 2A, 2B, in collaboration and led by the Gates Medical Research Institute. And that's the cetazolid, as well as a DPR1 inhibitor, which is a non-covalent inhibitor, TBA7371. And as you probably know, we have a number of phase 3 trials. NIX-TB was the one that I will describe to you, as well as Xenix. NIX-TB was the trial that allowed us to register Pertominate in this regimen for extremely drug-resistant TB. And the Xenix trial was a follow-on, and I will describe the rationale for that follow-on. And at this point, the enrollment is completed, and we're in the process of evaluating patients. And then, of course, Simplicity TB, which is really for a pro-drug combination that we had hoped to significantly reduce the treatment duration. But all of this is really not possible without all of our collaborators, and all of these are done with our collaborators. They are listed at the bottom of the slide. Sorry, it's a little bit too small, but it just gives you an idea of the number of collaborations we need to be able to support a portfolio of this size. And it's not just this collaboration, these number of people that we work with to advance these molecules is also not enough. And in fact, we benefit from being part of a consortium called the TB Drug Accelerator that is organized by the Bill and Melinda Gates Foundation. And through that, we have access to a tremendous amount of new technologies to be able to assess our molecules and progress them, as well as have access to the molecules of all the other members of the consortium from pharma or academic groups or institutions that we can then test in various regimens with, again, on the ultimate goal of getting a regimen down to 2 to 3 months. So let me first really describe to you the Xenix regimen that was approved last year through both at the FDA and the EMA and in India. And it really offers an incredible advance of achieving a treatment that is 6 months with less than 750 pills. And you compare that with the typical XDR treatment, which was greater than 18 months and 14,000 plus pills that needed to be administered over a long period of time with a very low success rate, as I pointed out earlier. And the main thing that the mixed TB did really proved is one of the key things is that it is important to discover a novel regimen, to work on a novel regimen, and also the fact that treatment of drug-resistant TB does not have to be long. It can be 6 months. It can be just as much as the standard of care for drug-sensitive TB. So that was really an important lesson for us that really proved that our strategy of starting with novel compounds to create novel regimens could be fruitful and ultimately be adaptable to all TB patients. So the Xenix TB, the results of them were published in the New England Journal of Medicine back in March of 2020. It had 199 participants in that trial. 71 of them had XDR TB, 38 had MDR TB, which were treatment intolerant or non-responsive MDR TB patients. And as you can see, the results after 6 months of follow-up, after treatments stopped, we had a 90% success rate with favorable outcome. That's a clinical resolution 6 months after therapy, which was quite significant compared to previous treatment regimens. And here I'm showing you the follow-up, 24 months follow-up of the Xenix TB regimen after the first 6 months follow-up. And the outcome did not change really as far as the efficacy of the regimen. The one thing that we wanted to note, or I wanted to note in this during this slide, is the follow-up of the side effects of this regimen, which was really mostly the peripheral neuropathy associated with the linezolid use. And that was also assessed during the follow-up serially with a standard symptoms rated from 0 to 10 using the peripheral neuropathy symptom questionnaire. And we then explored the fate of those patients, first at baseline and then following the complete follow-up time frame. So from an efficacy perspective, again, 109 patients were enrolled. We're all surviving patients except for one withdrawal completed the full course of therapy. At the primary endpoint, I pointed out to you that we had a 90% success rate or favorable outcome. And after the primary endpoint, one participant relapsed after treatment and one was lost due to follow-up. And after 24 months following the first 6 months follow-up, we continue to maintain that success rate and 88% favorable outcome independent of sex or HIV status. So that was quite significant. Now let me turn my attention a little bit to the adverse events, which really the main adverse event that would lead ultimately to either a linezolid linezolid dose interruption or reduction was due to peripheral neuropathy. And as you can see on this Kaplan-Meier graph, that the neuropathy really starts around the 3rd month of treatment. And here's a table showing you the different groups of people who developed peripheral neuropathy relative to dose and scheduling. So we had a 600 milligram linezolid twice a day dose, a 1200 milligram linezolid dose once a day. And as you can see, the participants who completed treatment were 40 in the BID dose and 63, total of 103. The participants received full uninterrupted 26 weeks of linezolid were 37 out of the 103, so about 30%. And the participants receiving full uninterrupted 26 of linezolid at 1200 were 16, so a little bit lower than the BID dose, or that any dose. And the mean linezolid duration in the treatment was about 23 weeks. And as you can see in the bottom, those are the number of people who developed neuropathies and saw them at baseline that we measured and then followed up after 24 months. And after the baseline, at baseline, 84 had none, of course. And after baseline, 27 had none. And then on the scoring function with a maximum mild to moderate symptoms, 34 had symptoms. And then in the maximum severe, 23 had symptoms. And after 24 months after follow-up of the treatment, as you can see in the mild to moderate, 29 had no symptoms. 3 still had some mild symptoms, mild to moderate symptoms. So, so you had some significant reduction and amelioration of the symptoms. And in the maximum severe, 13 resolved, and 7 continued to have some mild and moderate symptoms, and 1 patient continued to have some severe symptoms. So, in conclusion, we have a simplified, shortened regimen of all oral therapy for highly drug-resistant TB. And this was shown for even after 2 years of follow-up after the end of the treatment. But then, as I showed you, the peripheral neuropathy from linezolid was very common, manageable, and the symptoms improved over 24 months of follow-up. But then the question comes for us: can we manage the peripheral neuropathy through some optimal dosing and scheduling of linezolid, and hopefully reduce the number of cases of adverse events and minimize it significantly? And here we relied on our preclinical data. And the preclinical data that we used to justify our strategy is the relapse model that has been developed at Johns Hopkins University in the labs of Dr. Eric Nuremberger. And before I go through to some of the data, and you will see more data of this nature in table format, I just wanted to briefly tell you about the scheme of this relapse experiment. The mice are dosed 14 days prior, they're inoculated 14 days prior to treatment. And after 14 days, the mice are starting to be treated and followed up by at 1 month at a time. So month 1, month 2, month 3, 4, and 5. In this model, the standard of care cures the mice in about 5 to 6 months. And the strategy here is that at every time point, 15 mice are held for 3 months without treatment and then sacrificed, and then looked at the amount of bacteria in their lungs. At every point in time, let's say at month 3, if 15 of those mice do not have any bacteria in their lungs, so they are CFU-free, bacteria-free, then they are declared cured. And that will be the time frame for cure for that regimen in mice. So in the next slide, I'm showing you how the duration of linezolid could affect the sterilization activity of the regimen, especially in the bedaquiline pertominant linezolid regimen in BAL-C mice. And as you can see in the table, and if you can see my cursor, At the top, the regimen is the standard of care, which we compare to bedaquiline and pertominate to understand the effect of linezolid in this row, where we add linezolid to bedaquiline and pertominate. And as you can see, a 3-month therapy of bedaquiline, pertominate, and linezolid. At month 2, 6 mice still relapse, so 40% relapse. And at month 3, there are no relapse. So these mice are cured after 3 months of therapy with bedaquiline, pertominate, and linezolid. In the bottom 2 row, which was really interesting to us and exciting to us, is that if you stop linezolid after 2 months of therapy with this regimen, so you give 2 months of bedaquiline, pertominate, and linezolid, and then the last month you give only bedaquiline, pertominate, you still get the same relapse rate, meaning no mice relapsed at the 3rd month. Even more interestingly, you can give linezolid only for 1 month in this experiment, in this mouse model. So 1 month of the 3-drug regimen, and then 2 months followed by bedaquiline and pertominate. And in this case, again, we see no relapse in the mice. So that really formed the basis of the Xenix trial. And in the Xenix trial, we wanted to understand the effect of reducing the dose of bedaquiline, but also shortening the time frame of treatment with linezolid. So as you can see, there are 4 arms to this study. The first is the linezolid at the full 1200 milligram dose for 6 months. The second group is the 1200 milligram full dose of linezolid, but only given for 2 months. And the last 4 months would be just bedaquiline, pertominate. And the third arm is a reduced dose, 600 milligrams of bedaquiline, of linezolid for 6 months. And the final arm here again is reducing the time frame of linezolid with the 600 milligram linezolid to 2 months. The enrollment has been completed as of December of 2019, and we are currently following the patients and gathering the data, and hopefully we'll be able to present the Xenix results in the near future. And we're quite excited to see if our preclinical model also is indicative of the clinical outcome. So, talked a little bit to circle back again, because now I want to switch a little bit towards what's next and what are our efforts in developing even better regimens. So I hope I proved to you that the next TB was a proof of principle for us, that most resistance form of TB can be treated in the same time frame with as few of a drug as for the drug-sensitive patient population with comparable results. The next challenge, of course, for us is, and the most important challenge, one of the most important challenge is to develop one regimen that can treat all TB patients. We call it a universal regimen. So regardless of the resistance issue. And the initial goal for us, as I mentioned earlier, is to shorten the time frame for treatment to 2 to 3 months. And the long-term objective is a universal regimen that ultimately can cure patients in days to weeks, like any normal bacterial infection potentially that gets treated with a 7 to 10 day regimen. This, of course, has to be market driven, and we need constant feedback to be able to achieve these goals. So for the earlier stage research on how to achieve this, as I mentioned earlier, the most critical component is to have access to novel compounds that we can then put in novel regimens, test novel regimens at TB Alliance, especially with our relapse model. And to do that, we try to do it in 3 different ways. We first thought that we could apply the same strategies pharma companies do and see if we can capitalize on known compounds, known compound classes that work, but have side effects such as the linezolid. And if we can do better, if we can optimize a regimen that could be then adapted to drug-sensitive patients as well. Following to that on that is really focusing on developing novel classes based on known targets, such as RNA polymerase, which is the target of rifampicin. As I pointed out earlier, rifampicin has developed quite a number of resistance in the field. Plus, it suffers from cytochrome P450 induction and therefore has potential issues with drug-drug interactions. And then the third, really, which is majority of our focus now, is developing novel classes based on novel targets. And here, through the work of a number of great laboratories, especially through the TB Drug Accelerator, number of novel targets have been validated genetically, so one can start really focusing on some of those novel drugs within novel pathways that have never been tackled before, in the hope of finding a regimen, the ultimate regimen that can be really short, at least a 2 months regimen. So let me first talk to you about really focusing on the bedaquiline, pretomanid, linezolid regimen, which I hope I showed you how effective it is, but at the same time could be fraught with adverse events that cannot be tolerated in drug-sensitive patients. So what's next, and how can we do better? The first order was linezolid. Can we do better than linezolid? Linezolid, of course, has the side effect that I mentioned to you. It has bone marrow toxicity issues, and it also, more importantly, has neuropathy, peripheral neuropathy issues that really can become very severe. So the first order of business for us was to develop a safer version of linezolid. And we identified and are currently developing TBI-223, which is a novel oxazolidinone that has similar efficacy to linezolid and significantly reduced inhibition of mammalian mitochondrial protein synthesis, which is believed to be the cause of the bone marrow toxicity. However, the link between that and the neuropathy is still to be tested in some of these novel regimens. And the bone marrow toxicity that we evaluated in preclinical models, we have seen no bone marrow toxicity in up to 3 months in 2 species. Currently, we're in a single ascending dose. We finished a single ascending dose in healthy human volunteers up to 3,000 milligram dose and was well tolerated. And currently, we are starting a an MAD, multi-ascending dose in healthy volunteers. But I wanted to point you to the table below to try to convince you that TBI-223 has relatively similar efficacy as linezolid, so that if it is as efficacious and is significantly more safe, it could replace linezolid in the bedaquiline, pretomanid, linezolid regimen. In the table here on the row that says bedaquiline 25, pretomanid 100, that's bedaquiline dosed at 25 milligram per kilogram dose, which is equivalent in mice to the human therapeutic dose. Pretomanid at 100 milligram per kilogram dose given once a day. And then following that is adding linezolid at 100 milligram dose, and then comparing it to O, which is TBI-223, at the same dose of 100 milligram per kilogram dose. And again here, as you can see in the table, in the columns, the last 3 columns, we follow the mice at different time points. In this case, 1.5 months, then followed by 2 months, and then 3 months. And focus your attention on the last column. As you can see, for bedaquiline, pretomanid, and linezolid, 7 mice relapsed. And with bedaquiline, pretomanid, and TBI-223, which is the novel oxazolidinone, 2 mice relapsed. So basically, this experiment proved to us that from an efficacy perspective, we could expect that TBI-223 could replace linezolid from an efficacy perspective. And if it is significantly safer, then would be a great opportunity to test that in drug-sensitive patients as well. The next one was bedaquiline. How can we do better than bedaquiline? Bedaquiline is a member of a dialquilin family of compounds, and we sought to look for a significantly more potent dialquilin, reducing the MIC significantly, and therefore reducing the dose as much as possible, and also widening the safety margin as much as possible. And here I show you two compounds that we've identified and have progressed to phase 1 clinical trials. One we call TBHA 587, and then the other one is TBHA 876. And again, I'm showing you the preclinical data that we have generated with these two compounds. And again, in the row that says B25PAO, here we've already replaced linezolid with the new oxazolidine TBI-223, and follow the mice up to 8 weeks. And in this case, at 8 weeks in this experiment, 2 mice relapsed. If we go to the new dialquilins, as you can see, the 587 at 25 milligrams per kilogram, you had no relapse at 8 weeks, so significantly more potent, significantly shortened treatment. And if you up the dose to 50 milligrams per kilogram, now you are sterilizing the mice at 6 weeks. 876, the second molecule, at 6.25 milligrams, again, we had 1 relapse at 8 weeks, but more importantly, at 12.5 milligrams, again, we are sterilizing mice at 6 weeks. So a significant improvement in the bedaquiline, pretominate, and oxazolidine regimen that really gives us cures in mice in 6 weeks time frame. And typically, we are seeing cures in the 6 weeks to 2.5 months in this model. Now, as I mentioned, both compounds are significantly more potent. And in fact, if you look at the MIC90 of these two compounds, it is at 3 nanograms per ml. And again, quite significantly more potent than bedaquiline. 876 has completed the SAD studies and was well tolerated up to the top dose that we tested, which was 400 milligrams. And TBHA 587 has initiated phase 1 single ascending dose studies through the IMI consortium in collaboration with a number of partners through that consortium. So we really have quite a lot of hope that this novel regimen could be shorter and significantly safer, so that could be adapted to a wider patient population. Now, there's another advantage of these two new dialquilins that I would really want to mention, because there is evidence that there's a bedaquiline resistance is emerging in bedaquiline treated patients as well as bedaquiline naive TB patients. Now, the bedaquiline resistance TB actually is really rarely due to any mutation in the enzyme itself, in the ATP synthase itself, but it's rather in RV0678, which is a regulator of efflux transporter. So we've tested our novel dialquilins against these mutants. And as you can see in the table below, I've listed bedaquiline at the top row, which has about a 30 nanogram per ml MIC90. But as you can see, against these various mutants of 0, 6, 7, 8, the MIC increases dramatically to 1 microgram per ml or slightly below 1 microgram per ml. And as you can compare that to TBHA 587, it is significantly more potent against the wild type. But as you can see, it still maintains quite a good potency against all the variant drug-resistant mutants in the RV0678 strain. And the same results are actually true for TBHA 876. And I just wanted to follow up with just how that actually really translates in vivo in our relapse model. And here you'll see that after 1 month of treatment, back up, sorry, it's animated. Okay. 1 month of treatment. This is against the wild type, bedaquiline at 25 milligram per kilogram in the red bars, and this is TBHA 587 at 25 milligram and 50 milligram. And you can see that even as a single agent, they're significantly more potent in reducing the bacterial burden in mice. And at 1 month, if you now add it to protominate and linezolid and compare that, bedaquiline and protominate and linezolid are in the red hatched bars. As you can see, the TBHA 587 at 25 mg and 50 mg are significantly more efficacious at even 1 month of treatment. Now, follow up to 2 months of treatment. As you can see in this graph, the blue lines are the regimens containing TBHA 587, the new dialcoholin, and as you can see, we achieve a significant reduction of the CFUs compared to bedaquiline in the same regimen. Now, what about the RV0678 mutants, and how does it behave against that? Again, at 1 month treatment, already we see that at 25 milligram per kilogram and 50, the new dialcoholin is more effective than bedaquiline. The standard dose is 25 milligram per kilogram. Even at 50 milligram per kilogram as a single agent, we are achieving a significant CFU reduction, even with the mutants of RV0678. And this is in addition of protominate and linezolid. Again, the red column with the hatched marks is bedaquiline, protominate, linezolid, and compare that to the addition of the TBHA 587 at either 25 mg or 50. Again, a significant reduction of CFU versus bedaquiline against this resistant mutant. And then month 2, again, we continue to the improvement. And here it's a bit more dramatic even. And this is the red again is bedaquiline, protominate, linezolid. And as you can see, the novel dialcoholins, which are significantly more effective against the mutant, the RV0678 mutation, significantly reduced the CFU burden in this experiment after 2 months. So again, we hope that this regimen can actually address a huge need and also be able to overcome any existing resistance that is out there for bedaquiline. So now I'd like to switch briefly to some of our midterm goals. And here we are really focusing on novel targets and novel pathways. And I'm just listed here in graphical format some of the key pathways that one could target. The electron transport chain is well known. That's the target of bedaquiline, for example, and clofazimine. Many drugs inside the cell wall biosynthesis, central carbon metabolism, which is really a critical component here. That a lot of organizations are starting to tackle that. Protein synthesis, very well known, of course, with RNA polymerase rifapentine, as well as the ribosome inhibitor, the oxazolidinones. And then protein degradation, which is really, again, a pathway that has not yet been targeted, and it really is a housekeeping pathway to be able to digest unwanted proteins, very similar to similar pathways that exist in humans. And what we are aiming as one of the pathway strategies is trying to really modulate the protein synthesis pathway as well as the protein degradation pathway as an integrated way of targeting proteostasis. And our hypothesis was that if we combine the targeting of these two, not only we don't allow the bacteria to generate protein, but we also don't allow the bacteria to get rid of unwanted proteins. So we hypothesized that by combining these two, we will significantly shorten treatment and sterilize and achieve sterilization much faster. Of course, for protein synthesis, we will be using our novel oxazolidinone to test the hypothesis, and we are currently developing novel RNA polymerase inhibitors that are not rifamycin-like and they do not have the same resistance issues as rifamycins. And in protein degradation, we focus our attention on the CLIP-C1, CLIP-P1, P2 complex. And for that, we are focusing our effort on some of the natural products, the modulators that are out there to develop either peptidic inhibitors or non-peptidic inhibitors. And was this hypothesis that we presented to NIAID and received this a Center of Excellence in Translative Research grant to be able to demonstrate the hypothesis and generate two novel molecular entities that we ultimately could go into preclinical development. And this is a collaboration with Harvard and University of Illinois in Chicago and Johns Hopkins, and support from Schrodinger, one of the best computational chemistry groups in the world, and Texas A&M, one of the best in structure-based drug discovery as well. So where do we go from now? I hope I gave you a little overview of the early years of TB Alliance and establishing the PDP model, and really thinking about a novel way of developing new TB drugs, which really focused on novel molecular entities in regimens. In the 2010s, this is exactly what we did, and we proved that through the Xenix regimen. But for us, what comes next? You know, we are, as I mentioned, have a number of opportunities that could result in shorter treatment, maybe 2 to 3 months regimen. But what about beyond that? And we believe that really to get further down, we need to explore immunotherapy, as the cancer fields has done, and leveraging artificial intelligence. And this is really where we are going to focus a lot of our effort over the coming years in the next decade. And by that, we have a two-pronged approach. For the immunotherapy, we had over the last few years already started testing the hypothesis by either looking at specific pathways that we thought would be important in shortening treatment, or at least in modulating the immune system to shorten therapy. And we are about to publish a paper on that should come out shortly, hopefully. But then we also wanted to expand that, and we wanted to expand that in a more widespread way. And we partnered with a company called Inmany AI, which is an artificial intelligence company that demonstrate their technology, their artificial intelligence technology in the cancer field by identifying novel cancer therapies, immune modulators, cancer therapies that they're currently evaluating in phase 3 trials. So we developed the same concept to try to come up with targets and pathways that could impact TB clearance and management. And we have now a very significant number of opportunities and targets and pathways that we can explore. And these targets, interesting enough, are not related, of course, to antimicrobials, and also fortuitously they have either a clinical compound ready to be tested or research compounds also available. So we're in the process of forming an advisory group that will help us select candidates from this list that we can then evaluate in our relapse model, either on top of the standard of care or on novel regimens that we are developing. And at the same time, I told you there are a lot of new targets that are being validated and identified through a number of collaborations, especially within the TB drug accelerator. And we have partnered with a new artificial intelligence company that screens compounds through a DNA encoded library screening. And this artificial intelligence platform is a Google designed platform, and should lead to identifying molecules against these novel targets at the outset with far better physicochemical properties and drug-like properties that could then hopefully accelerate and reduce the time frame and cost for discovering novel drugs against these novel targets. And this could also potentially be adapted to identifying a target in immunomodulation and screening against those targets as well. So finally, really, this is a graphic that for our vision that we are aiming at the midterm universal regimen that could be less than 3 months, but really the next paradigm should be a much shorter treatment duration of 7 to 10 days, and we believe that that can be achieved by adopting the immunotherapy component, or at least modulating the immune system. And I mean, clearly the patient experience for the future would be dramatic as far as the time frame of treatment and the number of pills that they would take. And this is really not possible at all for us without all the support that we get, all the mobilizing a tremendous global network of partners, number of trial sites, community engagement sites, number of donors that we have, the portfolio that we hope to expand well beyond 30 projects, and the stakeholder association. And with that, I'd really like to thank our donors, because they are critical to our success. And as you can see the list here, we are indebted to them and are able to continue our research and development. And finally, I'd like to thank you for your attention, and I'd be happy to answer any questions that you may have.
Wow, thank you, Nader, for such an excellent presentation. I'm actually excited to see all this great progress in development of new TB regimen. I was here imagining a world with a universal regimen that can cure TB in just weeks or even days. Moreover, a regimen that is accessible to everyone worldwide. I really thank you for this highlighting presentation. Ladies and gentlemen, we've come to the question and answer session for our webinar. Please, as earlier shown by us, by the first presenter, Astrid, keep the questions coming in through the chat, as she showed in the chat, and we are going to try and answer as many questions as possible within the time that is allocated to us. So please keep the questions coming in. If you look at your slides, at your computers, there's a slide that is instructing how you can submit your questions. So we are not going to unmute anyone. We shall be reading out the questions, and Nader will be in position to answer them. Okay. So I'll start with the first question that I've seen in the chat. Someone is asking if, Nader, if there is any data on development of resistance or even cross-resistance to Pritomanid, that drug, Pritomanid. Is there any cross-resistance or resistance that you know of, or any data showing that? As we wait for other questions to come in, you could answer that.
Right. There are some, of course, pre-existing resistance through the nitroimidazole series, but in the regimens that we have tried, we have not really seen any of that occurring, especially in the Xenix regimen. So what we're seeing is more resistance emergence for bedaquiline.
All right.
We continue to follow that, of course, as much as we can. Not only in the clinical setting, we also follow this in preclinical settings as well in our relapse mouse model.
Okay. The other question that has come in is, did you not see any serotonin effect or platelet decreases with linezolid?
We did not. We did not. The number of patients were small, though, so we did not. Really, it was the neuropathy and the bone marrow effect that we'd see.
Okay. Then, have you seen any reduced efficacy with ritonamide in rifampicin-resistant strain versus drug-susceptible TB?
I'm sorry, can you repeat that question?
Someone is asking whether you have seen any reduced efficacy with the drug in rifampicin-resistant strain versus drug-susceptible TB.
So no, so far we haven't seen in a lot of that is in preclinical model. Of course, pretomanid currently has been tested only in extremely drug-resistant TB and the MDR TB patients who are intolerant to the treatment. And simplicity TB, of course, is also now being evaluated for drug-sensitive and drug-resistant TB. So we don't see any difference in efficacy as far as pretomanid against the various drug-resistant strains. I hope that answers your question.
Okay, and then someone is wondering what your view would be of putting moxifloxacin on reserve for TB.
On top of what?
Putting moxifloxacin as a reserve for TB.
So the simplicity TB is evaluating that, and we know from the TB practical that that's also being tested on top of BPAL, and we are happy about that and are excited to partner with MSF for that. So that's being currently evaluated.
Okay, and then someone would be interested in knowing whether the best way to assess PK for new agents. Are lung tissue drug concentrations really informative?
We believe, at least in our preclinical models, that the lung concentration is critical, but also we also know through certainly the work at the TB Drug Accelerator, the work of Veronique Lacroix, for example, and Cliff Barry and others, and Anne Leonard at CSU, that it's not just good enough to know how much you have in the lungs, but also where in the lungs. Is the drug going into the cavity? Is it going in the outside of the cavity? How long does it take for it to get into the cavity, for example, if the disease is cavitary? So a lot of those parameters are really, really important at the end of the day. Not all drugs have to behave exactly the same way, interestingly, meaning not all drugs have to get inside the cavity or just be in the granuloma on the outside. And this is really why, again, the regimen is so important. The right combination is so important. But all of those factors really make an impact. And I think the addition of moxifloxacin, the latest addition of moxifloxacin, probably can attest to that, because again, the distribution of various drugs in the lung, in the disease area, in the granuloma, as well as the cavity, can have a significant effect in the time to sterilization.
Okay, and then someone is wondering, what do you think is the most promising target for TB immunotherapy? In your view, what is the most promising target for TB immunotherapy?
So that's a difficult question to answer yet. We are just analyzing the data that we have generated with Invenu AI. We have to weigh really the pluses and minuses of which component, which pathway we need to modulate. There are a number of one of the pathways that we ourselves have tried, for example, is the mTOR pathway, and that's the paper that's going to be published. But there are a number of pathways, a number of targets, and that really can have an effect, including changing macrophage phenotypes, for example. So it's hard to tell until we really have a group of experts that we can sit down together with and try to then focus on the top 2, 3, or 4 of these targets, and then we will be able to let everybody know what targets we're working on. So we will put that on our portfolio, of course, and be able to show everybody what we are working on.
Okay. And then can beta-lactams be of interest against TB?
Yes, I mean the work that GSK published a couple of years ago already on immunoperim, for example, really demonstrate the utility of beta-lactams. But as you know, that's oral beta-lactams are very hard to come by and efficacious against TB. These are injectables. I mean, our goal should be all oral therapy, of course, but there is work being done with our colleagues at GSK on developing orally active beta-lactams.
Okay, and then someone is wondering about how TB Alliance works in this. So do you review data on new targets and new compounds published by academic teams and use these as a basis for new collaborations of members to the TB Alliance?
Yes, absolutely. We rely as much as possible on work that is being done in the academic world as well. A number of them do approach us with their data. We review the data, and we typically either want advice on what next steps to do on their own, or they want to form a formal collaboration with us so that we can do some of the work, especially through our collaborations that we have for assessing the efficacy of the compound as a single agent, and most importantly, in a regimen with our colleagues at Johns Hopkins University. So yeah, and then we form a variety of different collaborations with academic groups, and we actually welcome them.
Okay, an interesting question here. Do you see any future for TB vaccine?
Yes, I mean, I think TB vaccines should continue to work, but I think the eradication of TB really is to come up ultimately and relatively soon with a really significant regimen that you can then take, within weeks and then be done. So I think, to me, that is the best way of eradicating TB.
Okay, and then Nader, for your this is quite interesting to know. Do you have any considerations for any inhalation therapy instead of oral administration, or easy administration of the drug?
Yeah, I mean, our goal really is oral. I know there's work being done in inhalation and a lot of new technologies for inhalation. Again, for us, we're aiming at the regimen treatment that's just simple rather than have multiple pills and remembering to do the inhalation at the same time, whether that's basically once a day or inhalation is once every few days. We want to completely avoid lack of compliance, meaning make it as simple as possible. For us, an oral therapy at the outset is the first choice.
Okay, an interesting question from people who have lots of HIV. So in the murine models, what specific bone toxicities have you observed with linezolid? And in relation to HIV, what is your view on linezolid use in presence of HIV?
Well, as you can see, in our preclinical model, we don't dose linezolid, we're just at the toxic level of linezolid as well. We can't look for neuropathies in the mice, unfortunately. And the only way to evaluate the toxicity of linezolid is to treat rats and higher species such as dogs, and to look for their bone marrow toxicity or any other unwanted toxicity. So that we do see. Linezolid is seen, and therapeutic window is significantly less than 1 for linezolid. So that we see with linezolid. And for the HIV, as you saw, we had a number of HIV patients also in the Xenix trial, so it really was not impacted as far as linezolid use. I mean, of course, there was interruption of treatment, but overall the patients tolerated and took linezolid to the end.
Okay, so in your journey in as you develop these new regimens, is there any consideration for other non-traditional approaches other than immunotherapy, such as phage-derived approaches, such as antivirulence, and so on and so forth? Are there such considerations other than the immunotherapy?
Yes. Certainly the antivirulence is an interesting concept, absolutely. And phage display also is a very powerful methodology. I think there are advances in delivery of phage display. That would be one of the issues that we would be worried about. We will continue to monitor advances phage display therapy as well. But our first focus is really the immune system.
Okay. Okay, Dr. Nader, you mentioned working on molecules that target RNAP outside of the Rifapentine pocket. Could you speak to that, or have you published some of that work about those candidates?
Right, we have not published any of the work yet. We're still in the discovery phase, but we have a series of molecules that are quite potent against the enzyme. They do not bind to the binding site of rifamycin, so they're really not compatible, if you will, with the resistant mutations that we've seen using rifampicin in the RNA polymerase. So currently we are optimizing the properties of the molecule so we can achieve maximal exposure in the animal models to see efficacy comparable to the rifamycins.
Okay, and then are you working on any long-acting molecules, like molecules that you can use as implants for TB? Interesting question, yeah.
Well, I mean, we do already have a drug of that nature. I mean, bedaquiline is a very long-acting. It continues to remain in the systemic environment for quite a long time. Our new diaryl-coenzyl analogs also have long half-lives. They're shorter than bedaquiline, as far as we know. I mean, the data is still coming in, but they are still quite long-acting, meaning they remain in the system even after you stop the therapy. I hope that answers the question.
Yeah. Okay, and then what are some of the ways you would want to see? What are some of the challenges that you face while addressing this kind of work?
Well, the challenge really is, the main challenge is as quickly as possible to come up with new compounds that we can test clinically. We're still very short in the total number of compounds because to come up with novel regimens, we need novel molecules. And if you know the success rate in developing compounds in the clinical development, in the traditional sense, its success rate is 1 out of 10 across therapy areas. TB, we don't know yet the numbers because we're still, just ramping up. But we need significantly more new molecules than what I presented and what others have presented or are using in coming up with new regimens. So a lot more effort, a lot more research, a lot more clinical development has to be done. And of course, with that, that's expensive. And we rely solely on donors. So that, again, is another challenge that we need to to ensure that we have enough funding, not just us, but the rest of the world as well. All the other groups that are working on TB research to be able to be funded well enough to come up with novel molecules that we can test.
Okay, and then what's your opinion on new rifamycin derivatives to shorten substantially the treatment duration?
New rifamycin derivatives?
Yeah, derivatives for rifamycin to shorten the duration of treatment, especially for drug-susceptible TB.
Well, for drug-susceptible TB, yes. I mean, that's certainly, certainly Rifapentine was one first example. Yes, I mean, RNA polymerase as a target is a very important target. It's one of the most sterilizing combination of drug target rifamycins and RNA polymerase. One of the issues with the rifamycins, of course, if we can get around it, is the potential for drug-drug interaction, which becomes especially important in the HIV population, for example. So if we can come up with analogs that are that really do not confer drug-drug interaction, just for drug-sensitive patient population, yes, it would be beneficial. But again, as I said earlier, our goal is a universal regimen. We want the TB patient to come to the to the hospital, to the doctor, and be diagnosed, regardless of whether it's drug sensitive or drug resistant, and be given the same regimen, the same short regimen, and then go home and be cured.
Okay, and then more about the regimen that you talked about. Do you have a good outcome with a regimen in patients that have advanced damage in the lungs? And how did you really select the participants for the regimen? Did you consider chest X-ray, damage, extent of damage using chest X-ray, and so on and so forth? How did you select them?
Yeah. I mean, for the Nix TB regimen, of course, this was really advanced, right? These patients have been treated a number of times, and they probably have cavitary disease, etc. But I can get you the exact, we can answer this exact question as far as how many were evaluated, etc. Through our clinical development group. So I will take that question back and make sure that gets answered.
Okay. And then about latent TB, what is the current status of attempts to latent TB?
That's a very good question. So for latent TB, we don't have a formal research program on latent TB yet. First thing, obviously, we need to really understand what target drives latent TB, or what really drives latent TB, and what target can we target, if you will, to be able to have an impact on latent TB. And latent TB therapy also has to be extremely safe. I mean, typically, as you know, for the current therapy, we're using very well-known drugs that with many decades of safety information that we use to treat latent TB. So really, at the outset, one should start with an approved drug with a long-term safety data that one can then use to treat latent TB. But ultimately, if we can find how to treat latent TB, even at the core level of biology, then yes, we could definitely have some good programs on how to treat latent TB.
Okay, and then do you think there will be a role for fast-acting drugs in more drug-resistant TB and XDR TB in terms of treatment? Fast-acting drugs that you talked about. Do you think there will be a role?
I'm sorry, the fast-acting?
Yeah, the fast-acting drugs. Do you think there will be a role in MDR and XDR TB treatment?
You mean the daroluconilins that I presented?
Yes.
Hopefully, yes. Yes, we believe that if these are just as effective, for example, against the RB0678 mutants, if they're out there in the patient population, we believe that that could be adaptable potentially to as a universal regimen. Yes.
Okay. And then how do you feel about MTB-specific RNA targets?
RNA polymerase?
Yes, RNA targets. What do you think? Could there be a role considering RNA targets as opposed to other targets? What do you feel about MTB-specific RNA targets?
Oh, I see. No, we haven't considered those yet. We are right now, because of the vast or emerging validation of novel targets genetically in vivo. Again, we can't do everything. Those are the ones we're going to focus on because they've been validated genetically, and we know they have an impact on sterilization.
Okay. And then let me ask this question because I also thought about it. What are the main pathways you're focusing on for host-directed therapies?
For host-directed therapies,
Yes.
we haven't focused in on on the specific pathways. We had focused some effort on the mTOR pathway, which I mentioned. But right now we are evaluating the different pathways that we want to evaluate, we want to target. So we are going to form, we're currently working on a target within a pathway that I can't talk about right now, but we are going to choose from the effort that came out of the Invenia AI effort with an advisory group to select maybe 2 or 3 pathways or targets that we can then really hit hard over by the end of this year.
Okay, and then lastly, this being a COVID-19 situation, yes. Of course, you can't go without a question on COVID-19. So how has the TB Alliance navigated COVID-19?
Yeah, we've been quite fortunate. I mean, a lot of our collaborators, as you can imagine, different parts of the world at different time frame, especially in the US, had to shut down operations to focus their attention on COVID. So because we have such a large network of collaborators, we were able to shift work, from one organization to another organization in a different part of the world and continue to generate data and be able to continue to make molecules and generate data. So for our preclinical research, we fell behind a little bit, but not significantly. So we were able to very quickly mobilize alternative collaborators.
Okay, there are so many interesting questions coming in, but time is not on our side. So I would like to close the question and answer session now, but I would like to thank everyone for the active participation. We have over hundreds of questions in, but the time cannot be enough for us to answer all of them. So at this time, I would like to call on Astrid to close this up. And thank you so much, Dr. Nader, for this wonderful discussion and presentation. I'm still waiting for that time when we shall have that drug that can cure TB in days and weeks. Thank you so much for this wonderful presentation. Over to you, Astrid.
Thank you.
All right. Thanks a lot, Lydia, for moderating the Q&A. And of course, thank you, Nader, for your presentation and for responding to that many questions. A little note for the audience right now, because I receive this question very often and some might have joined a little late today. Yes, this webinar is being recorded and you will be able to view the recording on our website in the next few days, and it will stay there indefinitely. And another question I receive very often is about certificates. So yes, you will receive a certificate of attendance. This will come in a follow-up email, which will go out in the next few weeks. In this email, please scroll all the way to the bottom and there will be a download link for your personal certificate. And now I only would like to announce our next webinar. And end of April, we will have a webinar about CMC and formulation for antimicrobials with Alexander Danilovsky from Celia Pharmaceuticals. You can already register for this webinar on our website as shown on this slide. And in general, I would encourage you to check in on this website regularly. We will also announce more webinars there very soon. And with this, I really hope that you enjoyed the webinar, that it was useful for you, and that I will see you again on the line very soon. Thank you, everybody, and goodbye.
Goodbye. Thank you.
Bye-bye. Thank you very much.